So good morning, everyone. Welcome to the international webinar series of Sri Akilandeswari Women's College. So Sri Akilandeswari Women's College is celebrating its Silver Jubilee year. So to commemorate the Silver Jubilee year, they are organizing a beautiful international webinar series to educate young minds and also the academicians of the people from across India and also some parts of the world. And as in the Silver Jubilee series, we have today the Department of Biochemistry is organizing a beautiful program with having two beautiful speakers with us. And we have Dr.T.PONRASU, who's a senior postdoctoral fellow from National Yunlin University of Science and Technology, Taiwan, and also Dr.S.RAJESH KUMAR, the chief scientist and associate professor of Saveetha Institute of Medical and Technical Science, Tamilnadu, India, who is joining for the session 2. So today there'll be two sessions. So first session we are starting now, and then after first session, second session, Dr. Rajesh Kumar will continue. And after the second two sessions, the end of the session, we'll be posting the feedback link and the feedback form for the participants where you can fill in for your e-certifications. And without further delay, so we'll just hand over the session to Dr. Batmalochana. So who's going to give the welcome address and introduce our first chief guest of the day, Dr. T.PONRASU. Over to Dr. Batmalochana.
A warm good morning to one and all. On behalf of Sri Akilandeswari Women's College, Honorable Chairman, respected Secretary, respected Madam Principal, Vice Principal, teaching and non-teaching staff, and our departmental colleagues, on my own behalf, given an immense pleasure to welcome our respected chief guests and other dignitaries present here today's biochemistry lessons, and also welcome to participants from various colleges and university staffs and our beloved students. It gives me a great pleasure to invite and introduce the resource person for the today's biochemistry webinar first session, Professor and Scientist Dr. T.PONRASU has completed his doctoral degree from University of Madras during the year 2013. His field of specialization is screening of medicinal plants for the treatment of acute and chronic wound healings. He has 12 years experiences in the field of project assistant, research assistant, social research fellow, postdoctoral fellow, and senior postdoctoral fellow in National Yunlin University of Science and Technology at Taiwan 2018 to till this year. He received more than 12 various fellowships awards and patents during the year 2010 to 2017. As in biological societies, has published more than 35 research articles in cited Scopus Index international journals and published more than 15 media coverage of the research paper in the year 2018. Has acted as delivered more than 2 special invited lectures and acted as reviewer for Nature and Biomed Research International Journals. He has guided and co-guided for PG candidates at 5 PG candidates at IIT Madras from 2016 to 18. He has also attended for more than 25 national and international conferences. The importance of today's webinar, nutrition talent and promoting good will for updating academic knowledge in the field of biochemistry and research for during the pandemic period. Once again, welcome you, sir. Thank you, sir.
Thank you, ma'am.
Thank you so much, Dr. Batmalochana, for your warm introduction. And it's glad to see that the awards and the recognitions received by Dr. PONRASU. So it's been showing this academic excellence and also on the chosen topic. I'm sure you're going to give the beautiful lecture today, which will benefit most of the students and also most of the academicians over here. And I request Dr. T.PONRASU to take over the session.
Yeah. Thank you, sir. And thank you very much, ma'am, for the nice introduction about me. It's been a great pleasure for me to be a part of this Silver Jubilee webinar of organized by Sri Akilandeswari Women's College, Wandiwash. Before I'm going to start my session, I would like to thank the chairman and founder of this particular college, Mr. B. Munirathinam, and secretary, Mr. M. Ramanan, and principal of this college, Dr. S. Maithili, and head of the department, Dr. K. Batmalochana, and coordinators, Dr. M. K. Vadivalaki, and Dr. J. Vadivelu for inviting me for this particular Silver Jubilee webinar and this particular opportunity to deliver my experiences. Almost last 10 years I'm working in this particular field. I'm so happy to part of this particular webinar, and I'm due to the time constraint, I would like to start directly to the session now. So my topic is today the recent advances in wound dressing biomaterials for diabetic wound care and management.
So you can share the screen, sir.
One second, yeah.
It's not.
Why I have chosen this particular topic is India is, as we all know that India is having more number of growing diabetic populations nowadays. And among the diabetic populations, around 25% of people have immense risk for the lower limb amputations and chronic wounds, and leads to the amputation and loss of life also. So this particular critical conditions made me to start this particular area of research. I'm lost almost 1 decade and I'm continuously focusing on this particular field. So I'm very happy to share recent trends and advances in the wound dressing materials who have been developed from all over the world, and various scientists to improve the particular the wound non-healing diabetic wounds for the clinicians as well as the scientists. So before I'm going to start my impact on the major advances in this field, I would like to give a brief introduction about this the skin. We all know that the skin is generally known as the largest organ of this body, and despite it has some disputes, but we all know that skin is an important protective layer for the body, and it consists of 3 important layers. Starts with epidermis and dermis and kyphodermis. So epidermis is a topmost layer. It protects the body from external pathogens and external environment. It mainly consists of keratinocytes and melanocytes. Keratinocytes involves in the maintaining of the fluids in the skin, as well as melanocytes giving the color and the tone of the skin. And after that, it consists, the second layer is an important thick layer that is called dermis. It consists of major important proteins, skin proteins like collagen as well as elastin. It gives the integrity and functions, and it has an immense role on the skin anatomical structure. And finally, it consists of the hypodermis. It has fatty lipids as well as all other proteins which can be a part of this particular skin. So I don't want to take much time to elaborate about this skin. And this is a normal anatomical structure of the skin, and it is very important, and we should understand the basic nature of the skin to go in detail about the wound healing conditions. So next, the wound healing. Wound healing is an important thing. So it's a normal physiological response to the injury. So the wound is defined as a functional disruption or anatomical disruption in the skin, and it may be caused by the physical, chemical, thermal, or any immunological insult. So wound healing is a fundamental response to the tissue injury. It starts with 4 different highly complex overlapping phases. Starts with hemostasis and inflammation, proliferation, and remodeling. So hemostasis is the first phase, which involves the blood clotting mechanisms. And then it starts with inflammation. Inflammations will be a crucial part in normal and diabetic wound healing. So inflammation, in this inflammation part, the cleaning of the wound surface is mainly occurs due to the presence of neutrophils and macrophages. Macrophages precedes the phagocytosis of the unwanted things at the wound site, and then it helps to secrete numerous growth factors and other cellular modalities to start the proliferation of the skin. And this is a 4 interrelated overlapping phases is mainly important for the tissue regeneration. So the wound healing phases starts with hemostasis. It mainly involves the vasoconstriction, platelet, the plug formation, and degeneration and blood clotting. This is occurs immediately at the after the injury, and then at inflammation. Inflammation it's a main important plays an important role in normal as well as the diabetic wound healing. And proliferation phase is major part, which consists of formation of the major skin protein and angiogenesis, which further helps to provide enough nutrition and gaseous support to regenerate the damaged tissues. And finally, remodeling, that the collagen, the imbalances and balance of the collagen molecules, which will occur at the site of wound surface, and reepithelialization also covers the wound surface to repair the damaged tissue and get to the normal physical and anatomical functional integrity. So this is this particular slide will show you the cellular molecular mechanism of the wound healing phases. So here you can see the inflammatory phase, which is very essential in the diabetic condition as well as the normal wound healing conditions. Why I'm continuously focusing on the inflammatory phase and proliferation phase is mainly these two phases are defective in diabetic conditions. So these are the different types of cellular and molecular events which mainly occurs in the normal wound regeneration process. So as I said earlier, these are the mechanisms of different cellular events which mainly occurs during wound healing condition. So when we start to repair the damaged tissues, we need a proper and particular material to regenerate the tissues at the wound site, and it to protect the external trauma and environmental further damage, and it should be a natural tissue to regenerate. It should not be harmful to the tissues. So in that aspect, the wound dressing material should be ideal to regenerate the damaged site and damaged tissues. So generally, some of the important parameters should be essential when we take a material to treat the wound healing in any conditions. So those materials should have enough water molecules to dehydrate the tissue. If it is not possible to dehydrate the tissue, rehydrate the tissue, it will be critical to further trauma. So it should mimic the extracellular matrix, as well as it should be biocompatible, and it should, if it resembles to the natural soft tissue, it will be comfortable for the wound site. And generally, the material what we are going to use for the wound healing, it should be biodegradable and it should be non-toxic. And if it is easy to prepare and if it is cost effective, that will be affordable for all kind of peoples and all class of peoples to easily get the treatments. So when we prepare the materials, that should have important biological molecules or any kind of accelerating molecules to repair the tissues. That is, then it will be more advantageous to treat the wound healing. And when we prepare the materials, and when whatever the materials we are using, and if it has a angiogenesis ability, it's more advantageous to repair the tissues more adequately and rapidly. So it will help the produce the extracellular matrix and particularly collagen, and it helps to deposit the collagens at the wound site and accelerate the wound healing process. So my topic is, interestingly, diabetic mellitus and its complications. So diabetes mellitus, we all know that it's a mainly because of altered carbohydrate, fat, and protein metabolisms, and particularly for hyperglycemic condition. But this particular diabetes mellitus and its story, it has the great role of 4 famous scientists who are from the University of Toronto, Canada, who are actually started this particular discovery in a very detailed manner. So initially they started with the process, and it's a research initially started with the dogs only. So in the early 19th century, Dr. Frederick Grant Banting, who is a physician who had come from the arm injury from the World War I, who was more interested on the to find out what is the reason and what is the molecule playing a major role for reduce the blood glucose level. So who interestingly started with this research with Professor John McLeod, who's an advisor, who is an expert in the glycosuria and has published more than plenty of papers at that time, and who is an expert in that particular field. So he has granted the lab space as well as the one laboratory assistant called Charles Best. They were started with the experiment with pancreatectomy and ligating the pancreas duct to find out what is the product secreted by the pancreas, which is responsible for the to regulate the blood glucose in the stream. So they have started with pancreatectomy, and they finally find out that the extract, which is derived from the pancreas, which actually involved in the regulating the blood glucose, and finally they are not able to extract this particular compound. Then finally they got a help from the one more the biochemist James Collett, who helped actually to purify this particular compound, and they named insulin. For this particular discovery, they were awarded Nobel Prize in 1923, particularly for Frederick Grant Panting and John McLeod. Those two persons who were awarded Nobel Prize, and they were shared their Nobel Prize with Charles Best as well as James Collett. And this is a great story behind this the great research of finding the insulin molecule, which is responsible for the regulating blood glucose in bloodstream. So overall, we all know that the hyperglycemic condition is important the disorder which leads to the various types of complications in diabetic patients. It starts with microvascular and macrovascular complications, and finally, the major impact on lower limb amputation ulceration causes the loss of limb and amputation, and sometimes it leads to the death. So my focus is completely on lower limb amputations and ulcerations should be avoided through the proper treatment and proper materials to enhance their lifetime and healing the diabetic and chronic wounds. Normal wounds proceeds in the complex, highly overlated phases, starts with hemostasis to remodeling in a normal manner in normal wound healing conditions. But in diabetic conditions, due to the hyperglycemic condition, there is a lot of the consequences, as well as there is no clear exact pathophysiology is not understood by anyone. And there is a belief that there is a defective in two different phases. One is prolonged inflammation, as well as the defective proliferative phase. These two main phases is a reason for the delayed and impaired wound healing in chronic diabetic condition. So whatever the material we are going to develop that should arrest the inflammations initially, it should proceed in a normal tissue conditions, and it should enhance the proliferative phase as in normal wounds to improve the chronic wounds in diabetic conditions, and it should be affordable and cost effective. So in this aspect, the tissue regenerations initially started with a different type of methods, and to regenerate the damaged tissues and to restore its the functional and anatomical integrity of the skin. Earlier, in clinical trials and clinical aspect, they have started with autograft. It's nothing but transplanting the tissue from one side of the body to another side, and it has the problem is we cannot get enough tissues from the one part of the body to another body, and there is a chance for the contamination and further chronic conditions of the wounds. And then they started with allograft. It is getting the skin or any particular tissue from the one person to another person. That is also not possible because there is a chance for immunological rejection. So finally they started with the xenograft, that is from taking any kind of tissues or skin layers from one species to another species. And this also has numerous difficulties in the clinical treatments. So finally, they have decided to develop biological substitutes that is called biological materials, which will be a biocompatible, non-toxic, those generally called as biomaterials. In different type of biomaterials, the porous 3D scaffolds have an immense role to mimic the native extracellular matrix, which will be useful for the further regeneration of tissues or any organs. So in that aspect, the people and all over the world, the variety of the biomaterial scientists who have developed a different type of materials, they called hydrogels, films, foams, and different tissue adhesives. So all these materials generally based on the biological waste or biological materials. In that aspect, some of the materials have always great focus, like hydrogel matrix, porous scaffolds, and woven resin, and in highly advanced 3D printing constructs, and moreover, one of the important thing is nanofibers. All these materials are basically derived from the natural and synthetic biopolymers. Natural polymers always have an immense role due to the less toxicity and biocompatible for all the tissue materials. So this particular slide shows what are the current strategy involved in the treating the diabetic wounds as well as the normal wounds, and materials available in the different forms of material and their ability to treat the wound healing mechanisms. And all of these parts are normal wound healing cascades, which involved in the normal as well as diabetic wound conditions. And these are the major cell types who involved in all different types of wound healing phases. So the current trends of the treatment is the scaffolds. Scaffolds may be any type of polymeric based materials, and films and hydrogels and nanofibers. And initially they started with the Zucker diabetic fatty rat, which almost have the similar complications. What are the diabetic complications the human body has? So they started with a laboratory animal. There's Zucker diabetic fatty rats. It has an again it has an obesity based diabetic complications. So based on that, they have started with this male ZDF rat to transplant the all type of materials, whatever the materials they are preparing in the laboratory. Then they impregnated with the different type of stem cells, maybe adipose derived stem cells and mesenchymal stem cells to improve the chronic wound healing mechanisms. Then finally they transplanted into the wound site, and they found and analyzed different kind of pathological and immunological reactions to improve as well as to find out the efficacy of these particular materials. And what are the materials available? What are the basic requirements of any kind of biomaterial? It's the first and primarily we need the material with enough biocompatibility. Biocompatibility is more advantageous to use this particular material to regenerate the tissues and accommodate the cells and to migrate and proliferate, and finally to repair the tissues at the particular site. So, and it should also be what are the material? It should also has a role on biodegradability, because it should be biodegradable. Otherwise, it will be a complications again in the either topical treatment or transplantation of the site. So, based on that, we should fabricate or architecture that particular biomaterial. Those should have enough porosity, because porosity is very essential of that wound resin material. It's mainly because porosity can have an immense role to deliver enough amount of oxygen as well as nutrients to the all type of cells to grow without any disruption. So porosity plays a major role to support enough amount of nutrition and gaseous support. And finally, and all these hydrophilic scaffolds have immense role by holding the enough amount of water molecules and to rehydrate the tissue, and which will again improve the autolytic movements of the cellular events, and which will further stops the further damage of the particular tissue. And currently the materials are prepared with numerous type of growth factors, but when we use for growth factors, it has many disadvantages. The first is it's not affordable to all class of people, and moreover it's not having any sustained properties to hold and kept it for a long time. So the also lifespan of the growth factors are not feasible, and the cost effect also much more effective. So we cannot use those type of materials to treat the patients or any type of chronic wounds. So my ultimate aim, and the other people who are currently working on the preparing advanced materials, or the materials should be cost effective and simple, and it should not have any chemical cross-linking mechanisms. On further, it should have any biological active molecules to improve the angiogenesis as well as the enhance the collagen synthesis to repair the chronic wounds as early as possible as in the normal wounds. So the materials mainly should have a porosity as well as the cell biocompatibility that is generally assessed by the two different nucleic acid strains, which is FDA straining and DAP straining. And finally, that those materials should accommodate the cells to attach and proliferate at the wound site. Then if it has the pro-angiogenic property, it's a great role. It will play a great role on the repairing process. So overall, the biomaterials should have all kind of properties and with less cost, and if it is inexpensive to affordable for the all kind of people, it's a great pleasure to share these kind of materials for the further application. So far the material of choice is natural biopolymers and protein polymers. Natural biopolymers includes all kind of natural polymers like chitosan, fullerene, glycosome and glycon based polymers and all. Protein polymers generally includes the silk fibroin, keratin from human hair waste material, human hairs, and collagen from the animal waste, and gelatin also. So also the nanomaterials have an immense role on treating the chronic type of wounds, particularly recent trends involved in the graphene-based nanomaterials. And people are so far almost 2-3 decades they are preparing with silver nanoparticles to improve the contamination of the chronic wound site and further improving the wound healing process. And polymeric nanoparticles also in the form of liposomes or in micro vesicles to treat the chronic wounds. And growth factor loaded dressings are often available, but it's not comfortable and that is not affordable for all kind of people. So finally, the advanced thing is the materials, it has a property to release enough amount of oxygen at the site, played a major role, and it has some issues, but at the same time, when we are able to prepare a material with enough amount of oxygen releasing property, those can have a great role on treating the diabetic wounds. And 3D bioprinting materials also has a role, but it's not feasible to all kind of people. So I'm not going to talk all the major things and major advanced materials in detail. So I'm going to give us a few examples for each and every aspect. And the first, primarily, I would like to start with the nanofibers. Nanofibers have been prepared with plenty of natural and synthetic polymers, and it's predominantly alginate, chitosan, collagen, gelatin, silk fibroin, and keratin, even fullerene also. And synthetic polymers will have the great role, and particularly PVA will take major role in nanofiber materials, and polycaprolactone, polylactic glycolic acid, and polyurethane and polyethylene vinyl acetate. These are the major polymers has the different kind of roles for preparing and electrospinning the nanofibers. So electrospun nanofibers have a versatile space and having the ability to hold the molecules, whatever the biological active molecules, and it can easily release by a slow manner, and it improve the tissue regenerations rapidly. So in those point of view, they have all the people are always concentrating to electrospun the natural polymers or synthetic polymers combination to obtain a proper nanofibrous scaffolds with enough loaded nano or any kind of biological molecules to treat the chronic wounds. So the first one, I would like to start with hydrogels. This hydrogel is a simple and versatile material have been prepared by our lab, so my team. So I'm the part of this particular the compound. This paper has been published in Nature Scientific Reports in 2018, around age 17 in IIT. So this material is simply based on the chitosan molecule, which is cross-linked by sodium hydroxide, and which is loaded by the L-glutamic acids. L-glutamic acid has an immense role on is a precursor for the synthesis of proline and hydroxyproline, and proline is a major part of collagen molecule. And it's a collagen, it's a skin protein, which is very, very essential extracellular matrix protein for the repairing process. So this particular collagen is in triple helix form. The proline and hydroxyproline is a major amino acid which involves in the synthesis and to deposit the collagen at the wound site. So this L-glutamic acid hydrogels played a major role, and it particularly in the neovascularization and improved the diabetic wound healing. This is a great example for the hydrogel molecules, and this based on the laboratory animal studies only, not in a clinical study. And this material has a great role on neovascularization, and it improved the angiogenesis process, and through which it improved the diabetic wound healing early as possible, and particularly in 16 days is a normal condition. So another example is material based the wound dressing materials is also developed by my team, and I'm one of the part in this particular material development. So far the graphene oxide has been synthesized by the chemical methods, and we have taken the chemically synthesized graphene oxide to obtain these reduce the graphene oxide in sunlight. This sunlight process has been done in 15 minutes. This is so far this is a very fast and rapid manner of synthesizing reduced graphene oxide. This reduced graphene oxide have been in a nano form, and this was loaded in the isabgol. It's a kind of psyllium husk. This is a food material. It's not toxic, and I have loaded this particular reduced graphene oxide into the graphene isabgol seed. This is like a sponge material. It can hold enough amount of water molecules, as well as it is possess the ability to absorb enough amount of wound exudates at the wound site. Wound exudate in the chronic conditions can further damage the wound site, and it's a very complicated, unwanted thing in the chronic conditions. Which will further delay the impaired wound healing in the patients. So to avoid those type of complications, we have developed this particular isabgol-based material, which can absorb a large amount of fluids, wound exudates at the wound site, and deliver the graphene and then reduce the graphene oxide to improve the angiogenesis. The RGO already has been found that has an immense role and ability to neovascular angiogenesis to produce the enough amount of blood vessels and blood capillaries at the wound site. The exact mechanism is not yet understood, but it's the very least amount of nano formulation and nano amount of the reduced graphene oxide has a great role to improve the angiogenesis. So, and also it can have a great antibacterial role. Based on that, I have used this material to prepare this sponge-like isabgol seeds, and those played a major role and showed a great treatment and repairing process in diabetic conditions. So another example is the keratin. Keratin is a human hair protein. So the hair is a kind of waste material we are obtaining from the hair salon, and we are taking the minimum amount of say for example 5 grams of human hair keratins to obtain enough amount of keratin molecules by simple chemical degradation method. And finally, we have taken this keratin molecules to prepare a three-dimensional scaffold with gelling material of conjuglucomannan. This is also plant-derived natural polymer. It's a carbohydrate, glucose and mannose polymer, which is also biocompatible and non-toxic, and food material also. So these polymer and this particular polymer and keratin combinations had a great role. And apart from that, we have again loaded the avena sativa, that is also oatmeal extract, to further improve the diabetic wound healing conditions. This is one of the great example of interpenetrating network scaffolds, which is currently numerous type of scholars and research scientists they are developing with the different combinations of IP and scaffolds to improve the further manner to treat any kind of chronic and bone wounds. So these combinations helped and improved cell proliferation and attachment, and further it produced enough the feasible results on 16th day itself on diabetic wound treatment. So these are the some examples of the advanced wound dressing materials. Further, and recent developments are hydrogel based RT-PCR enzyme. This name can be a little bit not comfortable, but this material is very simple. They have taken the L-aspartic acid as an enzyme, and they have started with small and very less amount of copper sulfate, copper nitrate solution, and it's mainly because of they want to deliver the copper ions at the wound site to engulf the bacteria and avoid the contamination. And they have prepared with only L-aspartic acid and copper nitrate, and they have cross-linked through sodium hydroxide and form a gel. It's a transparent gel which can has a peroxidase activity to produce enough amount of reactive agent species at the site through which it can avoid the contaminations, and it can kill the bacteria at the wound site, through which it improved the cell migration proliferations. Generally, the caproion release will cause the major damage at the wound site, but this gel started to release slowly in the wound site, and the slow release of copper ions had a great advantage in the wound healing, especially in diabetic conditions. So the copper ions and its release improved the cell migration and proliferation, and finally improved the collagen deposition at the wound site, and finally they achieved the diabetic wound healing with the infected wound surface. This is recent development. And one more thing is the tissue adhesive hot glue. And this is a kind of polymeric based material. This is slightly modified with this is a polycaprolactone, the 4-star polycaprolactone. This is its hydroxyl group has been modified by N-hydroxysuccinamide, and they have taken this particular polymeric stick into the hot melt gun that will heat up to 50 degrees Celsius, and they have used two different range of polymeric materials. One is around 6 kDa and another one is 20 kDa. The 6 kDa 4-star polycaprolactone with polymer has melted and has solidified the wound site immediately, and it can has a very good adhesive property to hold the two edges of the wound site. So they have used this hot melt glue gun to deliver this particular protein gel in the as a wound extruding the wound surface, and finally they clipped and shielded this particular wound site, and they found that it has a great elasticity, and it's having protective and biocompatible at the wound site. This is also one of the recent development, but all other hot glue or all other glues has some disadvantages like flexibility, elasticity, and mechanical strength. But this particular this 4-star polycaprolactone N-hydroxysuccinamide glue has a great role because it's solid melting at the temperature of 39, which is around body temperature, and it's solidified immediately at the wound surface and produced enough amount of results and healed the wounds successfully at the wound site. And finally, this is one of the recent development of exosome-laden oxygen-releasing antioxidant and antibacterial hydrogels. This is simply they have prepared a polyurethane. It's a kind of polymer. They have loaded ascorbic acid as a antioxidant agent in the polyurethane gel. That is why they called as antioxidant polyurethane. They developed this particular cryogel, and they have loaded with calcium peroxide in particular concentrations to ensure the sustained release of this calcium peroxide for a minimum of 10 days. So this release of calcium peroxide and antioxidant potential of this particular polyurethane antioxidant gel has immense role to improve the angiogenesis and enough amount of oxygenation, and which further produced reactive agent species to arrest the further contaminations. And along with that, they have used exosomes. Exosomes is adipose derived stem cells produced on type of cells. This is a nanovesicles, and it has a great role on regenerating the tissues by enhancing the proliferation, migration, and synthesizing of collagen and deposition at the wound surface. So this is a recent development. The preparation of this particular oxo band is simple, but its ability to treat the chronic wound and at the site of injury showed an immense role, especially infected diabetic wound conditions. This paper has been recently published in Biomaterials. This is also from India, and this showed a promising results of how we can develop further any kind of nano or ultraspun fibers with enough amount of releasing capability of oxygen at the wound site. So when we load the calcium peroxide or any other hydrogen peroxide molecules and all, it has some critical issues while delivering enough amount of oxygen at the site. But this particular oxo band with enough amount of exosomes as well as the oxygen release capability produced enough amount of results in diabetic as well as the infected wound conditions. So these are all the examples of recent developments in the chronic wound healing treatments. So and also based on the available the natural polymers and other synthetic polymers, the people they have been generated different kind of tissue engineering materials. Starts with tissue engineering bladder and tissue engineering blood vessels and tissue engineering skin, and further trachea and tissue engineering corneal epitheliums. So these are the recent trend development of products by available natural bio substitutes called as a biomaterial. And what is the main advantages of the biomaterial wound resins is, for the particularly for diabetic wound healing. Diabetic wound healing is a global health care problem. As we all know that it's a very complicated thing for the clinicians to treat this patients, particularly with the high hyperglycemic condition. And so whatever the materials we are developing should be biodegradable and biocompatible, and it should protect from the further invasion of exogenous pathogens and other contaminations. And it should structurally, it should mimic extracellular native matrix. And these hydrogels and scaffolds have immense property of holding the moisture by absorbing enough amount of water by the hydrophilic matrices. So when we have the capacity of pro-angiogenic process as well as the regenerate the tissues at the wound site through these particular materials, also advantageous. And we should need a flexibility of preparation to obtain the desired material to treat the diabetic wounds at the site of injury and different type of the wounds. So finally, the biomaterials, whatever the materials we are obtained or we are trying to prepare through available basic natural carbohydrate polymers or a synthetic biopolymer combination by different techniques. So they are the excellent alternative wound dressing materials, and all these materials would be cost effective and feasible to everyone. So, and the preparation and fabrication methods will be significantly low when compared to the other materials like 3D bioprinting or ultraspinning, ultraspin nanofibers, and some growth factor loaded materials. So those materials can have the advantages, but the cost efficiency is very, very high. So we cannot affordable those materials in the clinically for the patients. So the cost effect of the diabetic wound treatments, it's increasing every year, and it's not affordable by everyone and each and every individual, particularly in India. So the future view, whatever the type of materials, it may be a hydrophilic hydrogels or scaffolds or nanofiber scaffolds, those would be biocompatible. Those would be derived from the biological substitutes, and it should have enough amount of porosity by preparing the bioproducts. So it should accommodate the cells in a feasible manner to improve the cell viability, and it should hold the cells and accommodate the cells for the further migration and attachment, which will improve the cell migration and cell proliferation. Finally, it can help in the tissue regeneration. And if the material, if it has the ability to produce enough amount of blood capillaries, that is also important thing to supply the enough amount of nutrition as well as the gas support to repair the tissues in a rapid manner. So the material properties are very, very essential. And the most important thing is it should be biodegradable, and biocompatible. So those are the essential thing in the primary objective. Further, if it has the angiogenic and antioxidant properties, those are the added advantages to regenerate tissues in the chronic conditions. Because chronic wound healing, especially in the hyperglycemic conditions, the tissue damage and the nerve damages are most common. So whatever the material we are incorporating in this nanofibers or nano scaffold materials, those would play a major role on avoid these kind of complications in the proliferative phase. So when we have a pro-angiogenic ability and oxygen releasing ability with loaded bioactive substances, those are the essential thing, and which can reduce the complications in the treatment. So the angiogenesis ability can be assessed by the tube formations in a human umbilical vein, the endothelial cells, as well as the CAM assay. This is a known in vivo method for understand the microcapillaries and blood capillaries formation. And finally, we can create the diabetic wounds by a known drug is called streptozotocin, and we can treat the wound surfaces by topical treatment of these different kind of materials to improve the cellular and molecular mechanisms to achieve the enough amount of angiogenesis, as well as the extracellular matrix protein called collagen deposition and its rapid regeneration. So these are the main aspects when we prepare the material to improve the chronic wound healing conditions, as well as the biological substances or bioactive substances we are going to load should be less cost effective or inexpensive, or then it can be helpful for the develop ideal wound dressing material for the diabetic patients. So with this, I hope due to the time constraint, I can't go and touch all and type of materials and its preparation and its application in detail. So thank you so much for your attention. And finally, I would like to thank for the organizing body of Dept of Biochemistry and the college, Sri Akilandeswari Women's College, Wandiwash. And with this, I would like to conclude my session. Thank you very much for your attention so far.
Thank you so much. Thank you so much, Dr. Thank you so much, Dr. Ponrasu, for your valuable session and valuable presentation. And I would like to request Dr. Rajesh Kumar from Saveetha Institute of Medical Sciences, the Chief Scientist and Associate Professor of Saveetha Institute of Medical Sciences and Technical Assistant, Tamil Nadu, to continue the second session, which is Art of Research Article Writing. So we are just glad to invite Dr. Rajesh Kumar, the Chief Scientist and Associate Professor, Saveetha Institute of Medical and Technical Sciences, Tamilnadu, India.
Yeah, thank you, sir. Thank you.
Hello.
Yes, ma'am.
Can you start?
Yes, sir. Yes, sir. Yes,
Welcome,
Doctor. Yes, ma'am. Dr. Wandiwash. Yes, ma'am.
Thank you. Thank you. Hello, everyone, and welcome to today's session. It's a pleasure to welcome our management, principal, vice principal, teaching and non-teaching staff, participants from various college and university. It's my honor to introduce today's chief guest, Dr.S.RAJESH KUMAR, Chief Scientist and Associate Professor, Nanobiomedicine Lab and Aquatic Nanotechnology, Saveetha Dental College and Hospital, Saveetha Institute of Medical and Technical Science, Chennai. Dr. S. Rajesh Kumar completed his PhD from MS University in the year 2013. He has more than 8 years teaching and research experience in the field of biochemistry, biotechnology, and nanotechnology. His work experience, he started his career as assistant professor in Adiparashakti College of Arts and Science, Kalavai, from 2013 to 15. From 2015 to 18, he worked as research scientist, School of Bioscience and Technology, VIT, Vellore. From 2018 to till date, he is working in Associate Professor, Department of Pharmacology, Saveetha Dental College, Chennai. He was awarded an honor of Young Scientist Award from DST and then Biozone Icon Award 2018, held on 27 February at IIT Chennai. Highly Contributed Paper Award in Springer. He has been invited as a guest lecturer in so many institutions. He also published 206 research articles in national and international journals. He attended more than 37 oral and poster presentation in national and international seminar, workshop, and conference. He guided for 9 PG candidates, 2 PhD candidate, and co-guided for 2 PhD candidate. Major achievement is project uptime 67 lakhs 70,000 from DST and ICMR. Research article published 206. Research articles accepted and consideration 35. Book published and accepted 3. Book chapter published and accepted 27. Patent field 11. Citations. Google citation 3047. H index 32. Scope of citation 1141. Research gate citation 2276. Audacious 36.7. Once again, I welcome to all. I pray Almighty for successful this webinar. Thank you, sir.
Thank you, Dr. Wandiwash ma'am, so for your great introduction. I'm glad to have, we have such an eminent speaker for the session too, Dr. Rajesh Kumar, who is going to give an address on art of research article writing. So thank you so much, sir. The session is over to you.
Yeah, thank you so much, Mr. Dinesh. And I would like to thank the management and Professor and Head Dr. Pathamochna ma'am and Dr. Wandiwash and Dr. Wandiwash. Shall I start the session?
Yes, sir. Please, sir. Proceed, sir.
Yeah, okay. Thank you so much. And the last session is very nice. I have attended the session. That is my research area, nanobiomedicine. Thank you, Dr. Ponrasu from Taiwan, and very nice session. And my topic is art of research article writing. Thank you, Wandiwash, for the good introduction. And I have published more than 200 research and review articles, and more than 20 book chapters in Springer and Elsevier publications. So I think I feel very happy to deliver this topic in research article writing. And then different types of research areas are there, and my research area is nanobiomedicine. So I will give some example in nanobiomedicine field. And then types of articles. First of all, we would like to know what are all the types of articles are there. First one is original article, review article. In that review article, systematic review book chapters are more or less same like work. And then letter to editor, correspondence are the short communications, case reports especially for physicians and dentists, and survey and extra, etc. And then first we have to think of what is the personal reason for publishing, why we want to publish our papers. First, we want to know what is meant by research, and we want, if you are a PhD degree, get funding, get promoted. And my point of view, publication is for my satisfaction as a researcher. Those who are finished PhD, those who have started their career as a PhD scholar, they are all coming under a scientist. So after if we are doing some research, what is the final one for your research? We want to publish our papers. See, nowadays, in our college, BDS students are publishing more than 3 papers per year. And then so many colleges, they are publishing PG papers, MSc, MTech research articles they are going to publish. And then most of the universities publishing paper for MPhil is compulsory, I think, nowadays. And then final one, PhD degree. We know that more than 99% of PhD scholars they want to publish their research articles in good journal. Research article as well as review articles and all. And for get funding, we want to have some particular research area. In that research area, mostly those who are scientists are planning to get some funding to develop their research and to develop the products and all. See, the last speaker published very good research articles in high impact journals, and he worked with very reputed institutions, and then he published very good articles. And his research area is very good research area. And for doing that research and all, we want some fundings. So if you want to get funding, you want to be a expertise in particular area. For that particular area of research, how we are going to prove I am a researcher in this field? If you are going to prove I am a researcher in this particular area, you must publish some research articles in that particular area. If you publish some research article in that area only, they will recognize you are a scientist in this particular research field for getting promoted. And the final one, why we want to publish? Main thing is, I always said to my students, publishing papers for our satisfaction. We want to get satisfied. Okay, I published this work in whatever the impact, whatever 0.1 to more than 10 also, if you're going to publish it, we want to get some satisfaction. So in my research area, I have published this research articles. Okay, that is the main thing, satisfying. However, the editors, reviewers, and the research community don't care about these reasons. Whatever the reason it may be, the editors and reviewers are expecting very good research article or review article for publications. See, in some journals, I have submitted many research articles to good journals. See, some journals, only 2 or 3 reviewers can review. See, in my personal experience, I submitted one of my paper to Journal of Nanostructures in Chemistry, Springer Nature. See, more than 7 reviewers reviewed my research article. My review research article is only 10 pages. I have received more than 3 to 4 pages of reviewer comments. See, like that also some journals are there. See, they are reviewing our research work. So we want to publish very good research work. See, if it is a positive or negative results, whatever it may be, see, keep it in mind, research is not a practical. Always think like that, because practicals always your expected result will come. In research, there is no expected results. Mostly we are doing research for expected results, but sometimes we will receive negative results, or negative in the sense we will get some new experience. See, for example, as a nano researcher, see, I have prepared silver nanoparticles by using neem extract. It shows very good results. Okay, it shows very good antimicrobial activity. See, the same, I have synthesized a silver nanoparticle by using guava leaves, but it shows very less activity, or the characterization part and all, it is not good. Whatever we have characterized, we did some activity. If it is a negative results, negative in the sense very lower activity or whatever it may be, we must publish it. See, people are asking why we want to publish these data. See, research work, data will be a data, whatever it may be. We want to publish. See, in future, some researchers are planning to publish their whether they are planning to do some research in Goa leaves, means they will refer that same like topics in Google, and if they will get that our paper, it may be we published, means they will get the data. Oh, okay, previously some people did some research in that area and the activity is not good. So what they will do, they will move on to some other plant extract. See, in the time, the chemicals, energy, time, that and all they are going to save, because based on our data, they will get some idea. See, what is the reason for publishing papers? Main thing, we want to do some research for our satisfaction. After our satisfaction, we will give it to our society. Most of the research we are going to do for our society only, because what are all the products we are using? See, from cell phone to toothpaste, see, for a toothbrush to some other product, car, bike, whatever it may be. In that backside of that products, some very good, very long research is there. See, based on the research only, they got so many failures, so many failures they got. After that only, they got some what are all the products nowadays we are using. So whatever it may be, we want to publish the data, whatever we get. See, think about why you want to publish your work. See, already I told for degree, for promotion, for whatever it may be. And have you really done something new and interesting? 4 important points. Okay. 4 important points that check the originality of the idea at the very beginning of your research. Very beginning, I would like to say, you want to read more and more research articles. Okay, that is the main point. We want to read. If you are planning to choose, I want to choose this plant, means that plant and plant based some biomedical application, means you must read about that plant. What are all the studies are there? Not only in your field of interest, you have to read top to bottom, you have to search that plant and what are all the applications of the plant. You must write one review article on that plant. Okay, then only you may get a lot of ideas. Before entering into research article, you must publish one review articles, because review article only you are going to get lot of knowledge, lot of ideas about the plant. Because while we know that we are not going to choose any new plants, we know that, because for that the different area of peoples are there. That's in botany, peoples are going to search, they only doing research and finding of new species. But like microbiology, biochemistry, biotechnology, whatever the applied science peoples are going to do, application of the plants. Mostly I'm telling that, and based on that, what are all the applications are there for that plant extract, we must collect research articles related in this area. If you are planning to do it, any application based things, you have to search. If you are planning to do anti-cancer activity of that X plant extract against lung cancer, you must collect research articles related in this area. Yeah. If you are planning to do any application based work, means you must do. See, for example, I told anti-cancer activity of that A or X plant extract against lung cancer, means you must collect research articles which is related to lung cancer. What are all the plant extract previously used against the lung cancer cell line? You must collect lot of research articles related to that. Mr. Dinesh, I think somebody muting.
No, sir. No, sir. No one is doing. Yeah.
Yeah, okay. I think I don't know why it is happening. Yeah, okay, then. If we are planning to do application-based study, we must collect research articles which is related in this area. And what are the plants peoples are used, and what is the connectivity of the plants, what is the family and all. We must collect lot of research articles. Then we'll move on to writing research articles. We want for every research article, we want one good idea. The idea will be from good review article, good research article collections only. Okay, that is the basic for doing good research. Choosing very good topic. So in that, for a research article writing, have you really done something new and interesting work? Okay, is there anything challenging in your work? The work directly related to current hot topic. That is difficult if you are planning to give a solution for difficult problems. How people are choosing the topic, means mostly in our scenario in Tamil Nadu or whatever, people are choosing topic based on the for applied sciences. They are choosing plants. Mostly people are doing research with plants, means they are exactly collecting plants which is available in this area. Availability also very important one, because if the PhD and all, we have one short duration, that is 3 to 4 years. Okay, so if we are planning to do availability of plants, we must do the application part will be a very useful one for the society. See, always before doing your research, you must think what is the useful thing for our society. Okay, always we have to choose. We want to ask some questions. If some common peoples are asking, what is the use of your research? See, we know that because we are all from one very backward area. So common peoples are asking, what is the application of your study? What is the use for the society? இதனால இந்த நாட்டுக்கு என்ன யூஸ் இருக்கு அப்படின்னு ஒரு கொஸ்டின் ஒன்று இருக்கு. See, always we'll do research is always we have to think what we are going to give it to what we are going to give it to this country. நம்ம இந்த நாட்டுக்கு வந்து நம்ம பண்ற ரிசர்ச் எப்படி யூஸ் ஆக போகுதுன்னு ஒரு கொஸ்டினர். Always you have to think that question. Okay, that based on the question we have to choose the topic, and based on the topic we are planning to do the research. Based on your research you are going to write your good research article. We are going to publish it in good journal. See, for the interesting research article, these are all the common things, which is title, abstract, keywords, introduction or background, materials and methods are experimental section, result, discussion, references, acknowledgement, conflict of interest. These are all common. Some journals are telling that background. Some journals it will be an introduction part. Some journals that experimental materials and method. And these are all the main things for the main subtopic for your interesting research article. How to write a good manuscript? For that, I told 4 points: preparation before starting, construction of an article, some technical details that need special attention, and language. These are all key factors. If you are planning to publish your paper in a good journal, always keep in mind preparations before starting, construction of our article, some technical details that need special attention, and language. For that language, very good scientist, always we are going to publish our papers in English language. So we have to discuss with some English speaking scientists, or else we have to write good research article that we will discuss with English scientist. English speaking, native English speaking scientists. So many of my research articles I have received this comment. They are telling, the editors and reviewers are telling that always you have to correct your research article with native English speaking scientist. Or else if possible, we always, most of the peoples are discussed with the English professors, those who are working with their institutions, and they are doing some corrections. Okay, Dan. And then title. Before starting this, we have to make one word file. Always in word file we are going to do, we are going to prepare our research article. In the word file, you have to make that outline for your research article. Outline, simply you have to write tentative topic, just tentative topic, abstract keywords. For in that during writing we will miss something, so we have to make some subtitles, and we have to make some tentative title. In that first, for every research article, people should see your title only. My suggestion is, because I want to say my suggestions, always you can give title for your research article in last, after finishing your introduction, materials and method, result and discussion, references, whatever it may be, because title is the major, and based on the title only people will guess what is the content in your research article. So always you have to give very suitable title in the last. After finishing your research article, you have to give it. So the title will be, because why I'm telling that means, for preparing your research article, you will refer more than 20 or 50 research, 20 to 50 research articles. Based on that, you may get some ideas. Okay, while we are studying some research article, you may get some ideas. Based on that idea, based on your research, what is the heart of your research? Based on that, you have to choose your topic, because the title will be attract the reader's attention. Just while when you are searching it in Google, just you are typing preparation of nanoparticles, green synthesis of nanoparticles, just you are typing in Google. In that, it shows more than 10 titles, 20, 30, 40 in different pages. It shows first, it shows topics only. So in that title will be attractive one. and check whether the title is specific and whether it is reflects the content of manuscript. So only I told, first of all, before starting your research article, after finishing your research article writing, you have to choose your topic. You have to give the topic to your whole research article, because in that content, you know that you only did the research, so you have some idea. So anyway, after finishing your research article, if you choose the topic, means it will be very suitable one. And because I told the topic will be a major thing, so we want to give some attractive topic will come after finishing a research article writing only. You may get, apart from that, will be a informative and concise. Don't give the mechanism of action of like that. Don't give the topic like that. Okay, that will be informative. Exactly, you are going to do anti-cancer activity against lung cancer, means the word of lung cancer cell line, and then anti-cancer, the word must in your topic. Okay, then it will be a concise, shortly you have to give. Okay, that will be an attractive one. And avoid some technical jargon, and then discuss with your co-authors. See, whatever for writing a research article, for doing your research, always not only one researcher is going to do any research. They are combined with so many researchers, and they discuss with their guide and all before publishing, before giving topic your topic to your research article. You must discuss with your guide, co-author, whoever it may be. Then the title will be attractive one. And then abstract. Abstract, that abstract, we know that two different types of abstracts are there. One is um, the short and the structured abstract. Structured abstract means in some journals are having some rules and regulations, and based on the journal requirements, it will be vary. In some abstract, we want to give some subtopics in that abstract. First one will be a background, small materials and method, small result, small conclusion. Like that, subtopics will give. And then another one side, the abstract will be a normal one. Normally, we will write more than 150 to 250 words. It may vary based on the journal need. So see, just you can think, abstract is a advertisement of your article. See, this is not only the advertisement. We want to give short and sweet abstract. Shortly, we know that what is mean by abstract. Abstract is the short form of your research article. In that short form, in the sense, what are the subtopics in your research article? The body of the research article, that will be introduction or background, materials and method, result and discussion, conclusion. See, that is the based on that, we have to make some short form of your research article. That will be a clear one, strongly influence whether or not your work further considered, and keep it brief as possible. While we are sending the research articles to the journal, first the reviewer will see the topic, and second, they will give the preference to abstract only. That abstract itself, they got some points. Okay, whether this research article is eligible, research article suitable to this journal or not, they will decide. And then they will see, for based on the journal topic also, that reviewers will say it is suitable or not. And then in that abstract, there is no comparisons. Okay, so many times I told, in that abstract, you have to include your result, not a discussion. Okay. And then keywords. Keywords, it is a label of your manuscript, and then only abbreviations firmly established, and check the guide for authors. That will be a number, label, definition, such as range and other special request. And because why we want to concentrate on keywords, in the sense, keywords mostly 3 to 5 keywords. Based on the journal requirements, it may vary. That keyword, while we are searching it, mostly we are searching research articles in Google. Just green synthesis of silver nanoparticles. Just we are giving it in Google means based on the keywords only that Google will catch your research articles, and just based on the keywords your citations may increase. Okay, based on the keywords the author will easily get your research, because why we are publishing, we want to get more citations. Okay, more citation in the sense so many people are referring your research article, then your research work will be somewhat worth, and it will be maybe useful to the society. So useful to the society in the sense how many people are going to refer your research article, they are all getting, they are all approving your research will be a good one. That is that will be that based on your keywords your citation may increase. So you must concentrate on keywords, and you have to see while you are referring research articles, have to see the keywords also. So many people are just studying their research article abstracts and all, they will not concentrate on what is the keywords measured. So many people, I know that. So just you have to see what are the keywords they have used. Okay. And then next, body of the research article. Introduction. From introduction, that will be a body of your research article. And before that, just my suggestion is, see, final, I told after writing your research article, finally you have to write title. Okay. After that, abstract. After that, keywords. That will be the three things are there, no? Abstract, title, keywords. You have to write it. Finally, you have to write it. Okay, finally, this is my suggestion. How I am following some, I have some rules and regulations. See, first always I will write materials and methods, because that the work will be your idea. So during your research work, side by side, you have to write your materials and methods in your notebook, because always I will refer, during your research work itself you have to write your research article writing. See, after that the materials and method you have to write result and discussion, and then introduction and result and discussion simultaneously you have to write, and then you have to write conclusion, and then references and all format changing that you are going to do. After finishing this materials and method, result and discussion, conclusion, introduction, after that you have to write your abstract. Final, I am telling that finally. After that, keywords. After finally only you have to give very suitable and informative title to your research article. This is the way I am following how I am writing research article. See, come to the point. See, introduction. Introduction is some journals they are telling that will be background. That introduction convince readers that you clearly know why your work is useful. See, in that introduction part, what is the problem? Are there any existing solutions? What is the main limitations? And then hope to achieve that and all you have to write. And then you have to give very recent information, very recent research in this particular area. See, if I am planning to write silver nanoparticles, I did research work on green synthesis of silver nanoparticles by using any plant extract. In that introduction part, we have to give very recent work on silver nanoparticles. What is the recent application? Because apart from previously I told, our research will be a current hot topic or novel work. So always, if this is 2020 means, always we have to give a reference from 2020, 2019, 2018, like that we have to collect research articles. Because in the recent research articles, you may get some recent information. That information that you have to add it in introduction part. And then the perspective that what's the nature of the journal based on some, I know that some researchers are writing research article to, they'll target particular journal in the sense, if it is a journal of photochemistry and photobiology means, they will write the introduction accordingly. The research work which is closely related to the journal. See, always See, my field of interest is nanoparticle research. So we are publishing papers in broad range of journals. We have colloids and surface and interfaces. And if it is anti-cancer activity, means that biomaterial research, nanotechnology, medicine, artificial cells, and like that, so many journals are there. We are choosing journals based on the applications. Apart from, if it is a nanoparticle research, we will choose the journals which is related to nanotechnology as well as related to the applications also. See, based on the journal, the introduction, small variations will be there. Because we have to add the point according to the journal needs. Because the reviewer, just while we are reading your introduction part, they may satisfied with your introduction or background part. However, hate improper citations of too many references relevant to the work. Don't add. I told that if based on your topic, if your topic is, I told, anti-cancer activity of copper nanoparticle, means in your introduction, you must add introduction to anti-cancer activity of nanoparticles. What are the nanoparticles previously used? Like that points you must add. What is the nanoparticle you are using? What is the recent advances of your nanoparticle in that particular work? That and all you must add it. Apart from, if you're planning to choose lung cancer cell line, means what is the recent research which is going in lung cancer cell line? What is the nanoparticles, if any research, if any nanoparticles which is used for lung cancer anti-cancer activity, that you must collect. Don't give a very old stories in your research article. Don't write old stories. You have to give proper points. Apart from justification, you must give. Okay, what's that? Justification. See, previously they did research like that. Why you chosen this particular topic? You must give the reason. Okay, apart from, you have to give the objective of your study. Objective in thesis, you know that you have one separate page for objectives to collect the planting, to study the characterization of nanoparticles, to study the anti-cancer activity against, like that you are giving them. That will be in a one sentence, like you have to make some very good objective of your study. Objective before that, you have to give, what is that, why you are choosing this topic and what's the advantages, that and all you have to give it in introduction part itself. And then never use more words than unnecessarily. Don't use the words which is not related to your research study or very old stories. See, if you are using some neem plant in neem or tulasi plant, means don't give the old stories. Tulasi 1,000 years before tulasi is used for the study. Don't give that stories and all. Recently, what are the research is going on tulasi? What is so biomedically they used tulasi for any activity, or if it is anything involved in any mechanism of action, like that points you have to give. Okay, don't write the old stories which is related to that plant extract, nanoparticle, whatever it may be. And don't mix the introduction with the result, discussion and conclusion. See, people are doing that mistake. See, in that discussion part, we are going to discuss with others results. That is discussion part. And people are giving the same, like in previous studies. Don't give discussion part in introduction. Okay, that and all we have to follow. And then the expression, people are telling, don't use the words like novel. Okay, we know that by using tulasi, thousands of papers are available in Google. So don't give by using tulasi. First time we are using tulasi. No, don't give words like that. Okay, see, some reviewers may accept. So from so many reviewers, the reviewers are researchers. That researchers will not accept that reasons and all. Why you are using tulasi, and tulasi is one of the most. Don't give. Okay, what is the point? That point we must add it. Okay. And the next will be a methods. See, already I told, I just for my researchers, I always refer, you must write methods in first tentative title you are giving it. After that, you have to write methods. See, for materials and methods, always while you are doing your research, that day itself, see, morning you have prepared plant extract means afternoon itself, you have to write it in your notebook. Always keep one notebook with you. If you are a very good researcher, must keep it. In that day itself, you have to write materials and method. Just you can see the chemical bottle. See, that is silver nitrate. What is the company that you have to write? That is the silver nitrate purchased from High Media, Mumbai. Dot over. Then what's the procedure you have followed? I have, what is the glass vase you have used? See, for the thesis and all, we want to mention the glass vase details and all. And for manuscript writing, just you have to give, these are all the materials which is purchased from this company. Company and country you must give. Okay, for a genuine, some peoples you must add. And then if you are using microbes in the sense bacillus subtilis means, you must give the MTCC number or ATCC number, you must give it detailed. That is the information, because if you are using animal means, you have to give the animal ethical clearance number and all, you must give it. Okay, see, that and all you have to write it during your research work. Because some people, we know that research articles, some people are doing 3 months, they will do research. After 3 months only, they are sit and write their research articles. That also not preferable. In my lab, for my researchers, I always said, if you are doing any research today, that day evening itself, you have to type your materials and method in the research article writing. Because we know that within 3 months, we forget what is the company. So within 1 month, the chemical bottle, some people after the finishing of that chemical, they will discard it. So you see High Media or Sigma, you may forget. So always materials and method, you must write that day itself. Then only it is easy to produce original and genuine data. And then in that materials and method must include the detailed information. And then don't repeat the details of established method. Okay, see, if you did a protein estimation by using Lowry's method means, don't write what is Lowry's method in Lowry's et al. What is the chemicals that are added, that was mixed. After that, we took reading. Don't add that established methods in your research article. Because so see, if you are planning to submit it in a very good journal means, the editor and all will be expertise in this area. So they know, and if you are if some researchers are planning to read your research article, they want to know about what is the advanced applications you have used. They don't want to waste the time with your established method procedures and all. And then if it's a new method, what's the method you have used, that exactly you have to write. And then reviewers will criticize the incomplete or incorrect descriptions, because that is also a reason for rejecting your research article, that some reviewers and all, if you are giving any false methodology and all, if they, because the reviewers are expert in that particular research area, while seeing the methodology and all, they find no, this is not a correct method, they are telling. Because you may forget, after 3 months of your research, you are planning to write your research article, you may change something, a false information, that is not something it was happened. You forget to write for 3 gram, 3 millimolar, some people write 3 molar. I know, I find a lot of mistakes like that. See, so many in our lab, we have given 1 millimolar, some people write 1 molar. If you are giving 1 molar means they'll write 1 gram. That mistakes and all happen. That the reviewers easily find that mistakes. Okay, they did the mistakes in that area. So that is also a reason for rejecting your research article. See, after that, materials and methods, you have to write results part. Result will be a representative result should be present. exactly what is the result you got that you had right. Exactly. Exactly. If you got peak at 427 nanometer means exactly you had right 427 nanometer. Okay. Don't write like 427 means you had right 420 or 430. Don't write exact results what you get, that you have to write it, because in my research area green synthesis of nanoparticles, that is not a chemical method, this is the green chemical method. It may vary for plants and all, so we have to write exact results. What are the results yours, you have to write it, that and all. After that, don't attempt to hide data the hope of saving it for later paper, and you may lose evidence to reinforce your conclusion. Okay. Uh, if it is anti-cancer activity, for that you did 4 or 5 assays, means you must give 4 or 5 assays in that research articles itself. That article itself you must give it, and you have to move on for submission of your research article. And use subheadings to keep results of the same type together. And then see if it is see if in my field nanoparticles, so I am giving examples in nanoparticles. See, preparation of plant extract, synthesis of nanoparticles, and I have characterized the nanoparticle by using different types of technique, in the sense UV visible spectroscopy, transmission electron microscopy, EDAX, and DXA diffraction assay. Means you must give the subtopics, and in that subtopic wise you have to give your results. And then in that result, for generally tables give actual experiment results, and the graph are often used for comparison of experimental result with those of previous works or with calculated theoretical values. And in that table, and see some people are in the result part, see they did antimicrobial activity against gastrointestinal pathogens, whatever it may be, they did the research. Means see, for antibacterial activity, they will give photos. First one. Second one, they will give tables. They will give graphs also. See, graph or table, any one is enough. Okay. Apart from, some people will do, see, for the same antibacterial activity, you will give 2 graphs or 3 graphs. One will be bar diagram, one will be line diagram for the same result. Don't give like that. That will be irritating to the reviewers. Why you want to give the, if you are planning to give table, don't add graphs. If you are adding graph, no need to give table. If it is a graph, any one graph is enough for a single values. See, for the same zone of inhibition values, they will give line diagram, and that is not preferable. Okay. Apart from the no illustration should duplicate the information described elsewhere in the manuscript. And then apart from if you got the results of different activities in the same, for the same antibacterial activity, you did different assessments. In the subtitle wise you have to give. One will be a protein leakage analysis, means separately protein leakage analysis. One will be plate assay, that result, that part and all you have to give it clearly. Okay. If any of your results are see, some journals are there, they will have only three figures, 4 figures, 5 figure. Means see, you have to separate in a single image. If it is, which is the results which is closely related, and the results which is going to give same meaning, means you have to give XRD EDAX, you have to give it in a single image, 1A, 1B. Like that, we have to prepare the images. The images, we know that in that basic mind, while we are seeing some research articles, see, just if you are reading any books, means just you will see the figures. We know that just in newspaper itself, if you will open it, just you will see the figures which is present in the papers. So always, for figures you have to give more importance, because that is, if you did any mistakes, people easily find it in the images. See, the reviewers and all, just they will see the figures, after that only they will read what you discussed based on that results. And whatever, just in the image itself they will find you did some mistakes in that. What is the size, shape, that is what's the elements which is present in your sample and all, they will find it based on the figure. So you have to give more importance to the, see, we were, all the subtopics are important. So for figures, more importance you must give. Don't do any mistakes. If it is line anywhere, graph means must give y-axis, x-axis, exact peaks. Apart from, if you are preparing, so many journals are asking for supplementary data. So many journals are asking raw data. So always keep your raw data in a folder. Always you have to have, if any single research article means, for that you have to have one folder. In that folder you have to give some subfolders. In that subfolders you have to keep your figures in original file. That will be Excel file or raw data. You must have it, because while if the journals are asking means, you have to give the all raw data also. And then figure legend of the figure should be brief, okay, and it will be a very clearly you have to give, okay, and then don't repeat the words in all the figures, okay. That for that also you have to read some research articles, you may get some ideas. And then discussion, already I told, discussion, what is results, what is data you got, that will be your results. What is the data others got is discussion, okay. If your nanoparticle shows peak at 420 nanometer is your result. If same like work others, so they got 425 nanometer means, that is discussion part you have to add, okay. Apart from in discussion part, if you are planning to discuss with any one technique means, you must give same like data. In that don't give antibacterial activity discussion in anti-cancer activity, don't do like that. Also for that only I told, always you have to keep subtopics, okay. Sorry, after that the huge numbers of manuscripts are rejected because of the discussion. In the discussion part you have to write very neatly. Based on the discussion, people will say you read some research article, you read some others research article, because that if you are reading others research articles, you may get some idea. For every research article you must read more than 50 research articles in your mind, okay. All the reference will be, you have to memorize the references what you are planning to use it in your research article. And then then if you read the research articles of others, and you may write very good discussion part as well as very good introduction part to your research article writing. The discussion corresponding to the results, but don't that related others, see, compare the published result with yours, okay. Don't ignore work in disagreement with yours, compare it, convince and read it that you are correct or better, okay. See, discussion, don't write some journals are having a separate discussion part, separate results part, okay. And some journals are asking result and discussion part will be in a single paragraph, okay. See, then our final one is conclusion, how the work advances the field from the present state of knowledge. Conclusion is what you are going to conclude about your research. Always think like that. Conclusion will be your own words, okay. See, for introduction part we'll refer some research articles, for materials and method part, for result and discussion part we may refer some others research articles, and then we will write accordingly. But conclusion will be fully your points, what you are going to conclude about that research, what is the futurology of your research, like that you have to write. So many people are doing one mistake in the conclusion part, they will write the summary. See, summary, see, some people are doing, they will repeat the abstract in the conclusion part also. See, don't do the mistake, okay. In abstract also one subtitle, the structured abstract, one subtitle is there, that is conclusion, okay. See, in that one question arise, that is, I already told, no, based on your research, what is the useful to the society? People are asking the question, what is the usefulness of your research? What is, how it is going to use it for the society? Each and every researchers, we are doing each and every research for our society only. Nobody will do the research for a single person. Okay, see, based on the question, for that question you have to answer what you are going to give it to the society based on your research. Okay, that will be your conclusion. Okay, and I already told, don't repeat the abstract. People should do summarizing your research work in the conclusion. Don't do. Okay, conclusion will be full and full your own points, and you should provide the clear scientific justification of your work. You have to justify your research work. See, I have prepared a silver nanoparticle by using this particular plant extract. It shows lung cancer activity. See, in that lung cancer activity, when compared to previous studies, how much your nanoparticle is having advantages, how much it will be, how much it is going to useful to the society, and how much cost you are going to reduce, how much greener method you are going to use, what are all the side effects you are going to see. So if we will take any drugs, means, see, nowadays the COVID-19, what is COVID-19 and related research are vigorously going in the worldwide. People are referring so many drugs, the chloroquine, and they're referring some more, so many drugs and all. See, for why that people are, sometimes they'll use the drug, and within 10 to 15 days they'll say the side effects. Each and every antiviral drugs are having so many side effects. See, we are doing research to reduce the side effects. We know that whatever the chemical we are using, see, even paracetamol also having side effect. Whatever that sugar tablets they are using, giberclamide, that is also having some side effect. See, birth bills, that is also having some side effect. See, like that normal chemical drugs are having this much side effect, means just you can think that drugs which is they are using for COVID-19 and all having so many side effects. So only they are referring, this month they are referring one drug, next month will be another one, another one, they are referring because of the side effects. Cost effect. See, as the developing country, we have to target, we always, the research is targeting for low cost. See, people, so nowadays they are using one washing machine, and if you are planning to develop one washing machine, means that will be better one when compared to the presently what we are using now. Okay, we want to reduce the cost, or you want to use any greener chemical, greener technology, they are asking the same questions only. See, like that, see, this and all, some suggestions, some questions, that for that questions we are we have to give the answer in conclusion part. Apart from that, that conclusion will be indicate uses and extensions. Based on that, if, see, some, all the research will not give a pakka solution to that problems. They have some extension. See, I have developed this drug. In future, we have to change it and we have to develop it. And based on the development, see, first time I asked some that why we are going to publish. We want to get funding. If you are planning to get funding, we have to do some preliminary research. Based on the preliminary results, based on that publication, I am planning to do this activity. I am planning to get funding. After that, I am planning to develop one good drug to that particular disease like COVID-19, whatever it may be. Moreover, you can suggest that future experiments. See, so only I told in that conclusion what exactly you are going to conclude about your research and what is the futurology of your research will be added in conclusion part. And then references, I told, see what are all the research articles you are referring in that discussion part, in that background part, that you are planning to use it in reference typically. And people are telling that don't use irrelevant publications to your research article. Irrelevant, and I told the old stories. 1987, 1997, they did some research which in that particular plant extract, but that is not relevant to your study. Don't use the reference like that. And always, my based on my experience, I will refer more than 20 to 50 research articles based on the need. I'll add it in my research article. If it is a review article, more than 50. See, in that review article itself, so many reviews are there, some critical reviews, exactly mechanism of action, only 20 to 20, 20 or 30 research articles only there, means we have to collect it. Exactly, we have to write what's the exact mechanism of action, like that we have to refer. See, we have to collect research articles which is closely related to your research. Okay, and don't avoid excessive self citations and the publication from the same region. We have to collect research articles in broad area. Okay, but this a plant means what are all the studies which is there in the plant extract? What are all the recent year? I told recent year references always we have to use. If you are planning to publish paper 2020, we must use 2020, 2019. You have to give preference to this last 3 years, last 5 years papers. Okay, like that we have to write. And then review articles. And for review article writing, if you are planning to do your PhD, you must publish on review article, then start your research. Review article may give lot of ideas to you. Okay, what are all the problems are there in your research that you have to find by using review article? What is the research gap? Research gap will be there in your particular research area that you have to find by using review articles. That for writing review articles, you must discuss with your guide. If you are a PhD scholar, if you are a postdoctoral fellow, you have to, that also mentors are there. That you must discuss with your mentor or reviewer, and you have to write one review article. This is a lockdown time. This is the time to write good review articles, because we know that we are spending more time with laptops. So this is the time to write good review articles. What are all the topics in your mind? See, previously 1 year, 2 year before, you think I want to write, if you are having some interest in particular area, so I want to write some review articles. This is the time to write your own review articles. Okay, that review articles also having some same subtopics like abstract aim, background, data available, key data, recent data, conclusion, futurology, and references. That is, these are all subtopics which is related to your, if you are planning to do review article, means that. The notes for publishing very good research article, that English language corrections, typo errors. Typo error means that protein means we will write PROTEIN. In that we will do PROTEI. During typing, so many people will miss so many things. Using units, see for MUL, so many students are doing the mistake is MUL means they will write UL. See, mu will be in insert option. Insert symbols. If you go, just you can see in word file, insert symbols, it will mu will be there. See, some people are thinking that they will write UL. That mistakes. Apart from L means that will be caps. ML means small m, capital L. Millimolar means small m, capital M. That we have to write. That see, the editors and the reviewers, just they will go through the research article. They will not read the research article at a time. Just they will go through, they will easily find that mistakes. That mistakes itself, they will find you are a initial researcher. If it is a good impact journal means, they will not give a preference to that basic mistakes what we are doing. So using of units and figures, already I told it will be a clear one. That is self-explanatory figures you must add. highlights, graphical abstract, that supplementary data and all. You must keep it in your mind. Apart from in ethics, say no to copy paste, and plagiarism is the main problem nowadays. Don't copy and paste anything. For that only, for initial researchers, always will refer. You have to write research articles, review articles in your notebook, handwritten. You have to see, read some research article. After that, you have to write your own points in notebook. After that, you have to type it, because we know that if any options are there, always we will use options. Just we will copy and paste. Don't do like that. That is not an ethical thing for good research article writing. Apart from, for a good researcher, plagiarism is a bad habit. So don't do like that. And that is the main ethics. Apart from, you have to give properly, you have to give acknowledgements properly, you have to give co-authorship. Don't give a guest authorship for your friends or anyone. If they are involved in your research, you have to give authorship and all, and always discuss with your co-authors for topic, for abstract. All the authors, you must do corrections and then move on to submission of your research article. And then if you have any doubts, if you want collaboration, you must contact me. And these are all the things based on my experience. I have given some idea to you for writing research article. That's all. Thank you so much. And thank you so much for the management of Akilandeswari Women's College and our HOD, and then the coordinators. Thank you so much. And then what next?
Yeah, thank you so much, sir.
Thank you, sir.
Thank you so much, sir. Would you like to answer a few questions, sir?
Yeah, sure, sir.
Yeah. So I'll just take a few questions in the chat box.
Yeah, okay.
A few questions from the chat box, and you can just answer the same. I'll just check.
Sure, sir.
Yeah. So can you please advise about journal those who have not those who are not having any impact factor but some authentication? How do we know it's good for our publications?
Yeah. Yeah, journals are having some ranking levels, sir. In India, that will be first level open access journals, and then UGC approved journal, and then Scopus indexed journal, PubMed indexed journal, and then Clarivative Analytics, that is Thomson Reuters journal. In that, so many stages, so many ranking level of journals are there. Based on your research, we must just, you can copy your topic and paste it in Google or PubMed, and you may get some research article which is related in your research area. See, just you can see, you have to download the only 10 research article which is closely related to your area, and then you have to choose that particular journal, and you can visit the journal homepage, and in that journal, how many research articles are there which is related to your field, and accordingly you have to choose your journals and move on for submission of your research article in the particular journals. See, apart from, if you're while you're seeing your data itself, you may find, okay, only 4 or 5 figures are there, which is so many. If you are having the data with research articles, more than 50 or 100 articles are there, like that means you must move on to normal journals. If your data, if your plant, if I am giving example, if it is a plant mediated nanoparticle, that plant is not used anywhere, that characterizations are very very good and very high, your activity, and you did any mechanism of action, means you must move on to more than 2 or 3 impact journals. See, like that data, you have to choose your journals.
Yeah, thank you, sir.
Yeah,
and how to avoid self-plagiarism of our own manuscript while PhD thesis writing?
Yeah, that is the main issue in plagiarism. For that, always we told that you have to read a lot of research articles. For each, see, if you we have to write 2 research articles for single data. That is easy to avoid. See, for just you can think, if you are having data which is UV visible spectroscopy, scanning electron microscope, transmission electron microscope, means for a single result you had to write 2 different discussion part. Okay, and then while reading some so many research articles which is related to this area, we'll avoid self plagiarism. See, that is the way we want to read more research articles. We have to write 2 research articles for a single data, but we have to publish any one data which is satisfied to you. That is the thing. That is the way to avoid self plagiarism, because we know that see, writing single research article is very difficult. See, as a researcher, as a PhD scholar, you must write 2 different discussion for single data. One is for research article publication, one is for PhD thesis submission.
Yeah, thank you so much, sir.
Yeah,
and is there any free software to check the plagiarism?
No, because in website so many plagiarism softwares are there. This is free. See, just you will upload your research article in that website. It shows 0% Turnitin. That is 0% plagiarism. It shows, if see, just to keep it in your mind, if your research article shows 0% plagiarism, means that website is fake website. That software is fake software, because even for Nobel laureate also it shows more than 5% plagiarism, even for Nobel laureate also. But just you can think, just you can upload it in any website which is present in that Google and all. It shows 0%. Don't think I prepared a research article with 0% plagiarism. That and all fake. All
Okay.
the Turnitin and plagiarism checking softwares are cost only.
Okay, sir. So what are the percentage of the plagiarism can be accepted? For example, you're saying 5% or 10%.
Less
So
than
what?
10% is accepted for all the research articles, which is more than all the impact, more than 10 impact also they'll accept. Less than 10%, because some words are unavoidable, because just you can and the like that words will come. So less than, if you have less than 10%, your research article is eligible to publish it in any journal which is with high impact or whatever the impact.
Okay, sir. Yeah, thank you. And while citing a paper, is it required to write the reference number multiple times inside the paper?
No need to write like that. Just you can refer. If it is you are going to give a number, means 12 means 12 in anywhere. If you are using the 12th reference, means in the introduction 12, in the discussion part also 12 you had said. One time is enough. In that back side, in the reference part, you had write one time, you had write. That is enough.
Okay, sir, fine. And the next
Okay,
question
is
is,
that, are you satisfied with this answer?
actually it was raised by the participants, sir.
Yeah,
So,
okay, right then.
yeah, because the YouTube live, there is many participants are sharing their questions.
Okay, sure.
So I'm just sharing to you on behalf of them, sir.
Yes, sir.
And can you please share some UGC approved journal websites, sir?
Yeah, in UGC itself, just you can visit UGC website, and then just you can write a UGC approved journals 2020 in Google. That one Excel file will come. You have to download it. You had to use it. That you have to download it from UGC website also. It will be there.
Okay, sir, great. And participants, if you have any questions, so please share in the feedback links in the chat box for the first session speaker and the second session speaker. So we will post the same questions to the speaker, so they will be ready to answer your queries. If you have any questions, so please post in the chat box.
And any doubts, I have given my mail ID and mobile number also. You can
yes,
WhatsApp
sir.
me. Give
Yeah.
me mail. I will answer it.
Yeah. How to create a graphical abstract?
Yeah. Yeah, for a graphical abstract, first of all, you have to write your abstract in your manuscript writing, and that abstract, you may get some idea. Okay, right, then we have to present. If you are doing a mechanism based study, means that mechanism part you have to make it as a graphical abstract. Otherwise, if you are just a topic, I'm saying anti-cancer activity of lung cancer cell line from any plant extract means you have to give just in for preparing a graphical abstract, you want some imaginations, just plant-like structure, just one arrow in that one bioactive compound. After that, lung cancer means like that lung cancer. We have lot of softwares, a lot of websites are there in Google. Just you can type graphical abstract writing or figures writing, image making. Simply, we have to give some arrows like that. We have to prepare. If it is a mechanism based, we have to give exact points, exact what's your major application and major preparation. Based on the points, you have to prepare graphical abstract. That will be easy only. We want to read so many graphical abstract which is present in the journals you are planning to publish.
Yeah.
Like that we'll prepare.
Yeah. And then one more question from Ms. Bhagavati Sivatanu.
Yes.
In UGC website, there are updating the approved journals every year by year. So
Yes.
whether it's valuable for upcoming years.
Yeah, that is, see, always think, don't go, see, my personal suggestion, don't refer UGC websites and don't publish your papers in UGC approved journals and all. You have to move on more than level. You had to publish your papers in Scopus indexed journal. You had to publish your papers in PubMed indexed journal. You had to publish your research articles in Thomson Reuters journal. If you are publish it, then it will be in international database. If you are publish your research articles in Scopus indexed journal means all Scopus indexed journals are mostly 99% of Scopus indexed journals are in UGC. See, year by year, UGC will change. But if Scopus and PubMed, Thomson Reuters, your database in international database. See, don't think, for my personal suggestion, don't target UGC indexed journals, UGC approved journals. You can target more than that. UGC only for India. Okay. Scopus, PubMed, Thomson Reuters, and all international level. So you can target more than that. Don't care about UGC CARE journals and all. You can target more than that. That is my personal advice to you.
Yeah, thank you, sir. And what is, can you just elaborate about H index?
Yeah, H index, there is majorly two different H index are there. One with Scopus, one with, what is that, Google Scholar. Okay. There, that is Google Scholar, they have Scholar Society, some group of people are working with that. In Scopus also, some group of people is working with that. That is, H index will show how much you are great scientist. Okay, that will be decided based on how many people are viewing your research article, how many people are downloading your research article, how many people are citing your research article. Based on this points, they will give H index to you. See, just for my H index, just you can type Shanmugam Rajesh Kumar in Google. Shanmugam Rajesh Kumar. You may find my page. Just you can click Google Scholar. My H index will be more than 30, I think. That will be based on how much quality of articles. Because quality articles, people easily download. They will cite your articles. So based on citations, viewing, downloading of your research articles, they will decide the H index. That is for Scopus also the same, like that procedure they are following. Okay.
Yeah. Thank you so much, sir. And
thank you.
Another queries. Does research proposal and research hypothesis is same or different?
No, entirely different. Research hypothesis, research proposal is what you are going to propose. For every research proposal, you have to write research hypothesis. Okay, research hypothesis is one of the subtopic which is based on the hypothesis only. You have to plan your proposal. Based on the hypothesis only, you are planning to do your research. For if you are planning to do any research in that particular topic, means you have to write on hypothesis. Okay, it will be possible or not. Like that hypothesis you have to write. Accordingly, you have to write your proposals. And the proposals for a proposal in the sense that is for a funding agency, research, PhD and all it. Okay, entirely different. Hypothesis is a subtopic of your research proposal. That's all.
Okay, okay, sir, fine. So participants, if you have any other questions, you can just post here. So
Yeah.
we'll be posting to the participants. If you have any other queries for the session 1 and session 2, so please share your queries if you have anything. So how to write the references, which type of format should be followed?
Each and every journal is having their reference style. In that author instruction, they clearly mention how to write references. So many reference types are there, Harvard references, and then like that, so many reference styles are there. That will be changed according to the journal guidelines.
Okay, sir. And the next question is, suggest some tools to produce significant results graph of our data that we obtained in experiments.
Yeah, commonly for a graph and all, you can use SPSS software, and then Origin software and all available in website net, I think. Apart from for easy preparation of data, you must use Excel file. In Excel files, lot of options are there. After if you are satisfied with the Excel file, then you can prepare your graphs and all by using SPSS. Just noted SPSS software,
Okay.
and then Origin Pro, and also many versions are there.
Okay, okay. And the former question was actually asked by the PhD aspirant, like how to write the references for which type of format should be followed. That was a PhD aspirant's question, sir.
The more than 90% of PhD thesis, they will write a reference. In that, citation will be name and all. Rajesh Kumar et al. If you are in your research article, single author is there, Rajesh Kumar 2019. If you have two authors, Rajesh Kumar and Dinesh 2019. If more than three authors, Rajesh Kumar et al. 2019. That citation will be like that, and the reference will be for alphabetical order. That full reference you have to write. That is most commonly used reference format for PhD thesis, and name you have to include in body of the content.
Okay, okay, sir, fine. So which journal is appropriate to publish paper for UG students?
For UG students, whatever UG student, PG student, MPhil, whatever it may be, it is based on your results, based on the data you have, based on the genuineness of your journals. See, mostly for UG peoples, they will publish their work in based on their guide. They will decide we are planning to publish it in UGC approved journal or Scopus index journal. That will be decided based on the data only, based on your guide work. If you want to do publish your papers in a good journal, just you can send me the title and abstract to my mail ID. I will suggest what type of journal is suitable to your work. I will suggest personally. You can send me mail. I will suggest you.
Yeah, thank you. Thank you so much, sir.
Yeah,
And thank you. Thank you all the participants who's been posting the questions. And so I'm just looking for if there is any other queries. So yeah, that's it, sir. So thank you. Thank you so much for your time, energy, passion which you have taken. So thank you, Dr. Ponrasu, and thank you, Dr. Rajesh Kumar, for taking your time from all the way from your schedule to educate the academicians and students of India. Also, we had some other participants from across the globe are watching the sessions. It's been good. We had more than, at a time, we had more than around 250 people plus watch through the live. And I guess it'll be more than 3,000 plus views totally. Maybe I'll just check at the end of the session. So thank you so much. And I would like to request Dr. And participants, the feedback link has been posted already in the chat box. And also the feedback link has been provided in the comment box, and it will be available until 5 p.m. Today. You can just feel aramse, and then you can just take your time to post the feedback link. And thank you the Sri Akilandeswari Women's College Wandiwash Vandavasi for creating superb sessions for the academicians and students put together by bringing various scholars and organizing daily international seminar by hosting different participants. And we were hosted on behalf of IQAC Commerce Department, and yesterday it was from the Maths Department, and today it was for the Biochemistry Department. And 20th, we have one more program from the Department of Physics. And every day we are glad that the Akilandeswari College is hosting a foreign speaker and also an Indian speaker to bring both expertise together in a single session. That is a great boon for the participants. So thank you so much, all the people and who have been watching the live for last 2 hours. And thank you all the people like Dr. Selva Kumar and Dr. Maithili, the principal, and the HOD of the program, Dr. Batmalochana and Dr. Wandiwash, for everyone who has been hosting the session. And I request Dr. J. Vadivelu from the Department of Biochemistry to proceed with the vote of thanks.
Okay, sir. Good afternoon to all. Honorable Chairman, throughout a line, Dear Muniratnam sir, most respected Secretary, throughout a line, M. Ramanan sir, former Member Syndicate of Trivandrum University, beloved Principal, Dr. S. Maithili ma'am, Vice Principal, Ms. K. Suguna ma'am, IQAC Coordinator of the Akilandeswari Women's College, most valued Chief Guest, Dr. T.PONRASU sir, Senior Postdoctoral Fellow, Taiwan, Dr.S.RAJESH KUMAR sir, Chief Scientist, Saveetha Medical University, participants of various college, principals, scientists, staff members, students, and my colleagues. It is my privilege to have been asked to propose a vote of thanks on this great occasion. I, on behalf of the Biochemistry Department, extend a very hearty vote of thanks to the honorable delegates who blessed us with their presence and took out valuable time of busy schedule. I also wish to express my gratitude to our principal, Dr. S. Maithili ma'am, for providing encouragement and kind support. I am very grateful to our management for giving us an opportunity to organize this webinar. Thank you. Thank you so much for one and all present here. Thank you, sir.
Yeah, thank you. Thank you so much, sir. Thank you so much for your valuable, valuable vote of thanks. And thank you all the participants. And it's been a great time having you and all the participants who have been watching the YouTube live. We are going to close the YouTube live sessions. If you miss the chat box, the feedback link, just go to the comment box. It will be available until around 5 p.m. today. Please take your time to just complete your feedback form. So thank you all. Thank you so much for your valuable participation.